The main work of my lab is split into three sections. Part of it is studying how organs naturally develop, and that's looking at embryology, looking at developmental biology and cell biology to understand how we go from a simple egg to the complexity of an adult. The second part is trying to use that knowledge to engineer new organs. So, for example, we're trying to grow kidneys in culture from stem cells, ultimately with the aim of transplanting for human use, although of course that's a long way away. And then the third part of the laboratory is working on synthetic biology. And for the synthetic biology, we're interested in teaching naive cells that don't know how to do developmentally interesting things, how to make patterns, shapes, forms, and so forth. The point of that is that we think we understand developmental biology. If we engineer genetic systems based on our understanding and put them into naive cells, if they work, then yes, we understood. If they don't work, which is the more interesting thing, oh, we haven't understood how all this works, back to the drawing board. Isaac Asimov once said the most exciting thing to hear in a laboratory isn't eureka, it's that's odd. And we sort of live for those that's odd moments. We use a variety of culture systems for growing cells and for growing organoids, depending on the question that we're asking. So if we're asking a system just of very simple cells, we'll often use 2D, as we used in this system. On the other hand, when we're growing complete kidneys, obviously we're growing them in 3 dimensions. We can do that without flow to some extent, because the organs that we work on, kidneys, are very tolerant of low oxygen. But now that we're starting to face the challenge of growing larger kidneys, we can engineer quite small kidneys, and our biggest challenge is to get them large. We're having to face the fact that we are going to need flow to be able to bring oxygen in, nutrition in, and most critically to remove waste products. And that's really the next stage of our research, and I imagine we'll be using the Quasi Vivo system as a starting point, and then depending on what it does for us, well, we're hoping to work with you, in fact, in developing improvements to that system. For many aspects of biology, anatomy is very important to function, and anatomy and physiology go together. Simple cells in 2-dimensional culture can do many things, but that whole idea of culturing cells that way was really developed for growing viruses. It's fine for that, but it's not very good at mimicking the interactions between different parts of a tissue. Particularly for understanding how organs work, there are different cell types present. You often have, for example, an epithelial tube surrounded by mesenchyme tissue. The 2 types of cells talk to each other, and if they can't talk to each other properly, you don't get normal function. Also, in a lot of organs, the tubes are filling up with something, or something's being removed from the tubes. That's the point of the organ. If the tubes are not tubes, if they're not closed spaces, then we're not mimicking normal physiology at all well. So for all of those kind of systems, I think it's important to use 3-dimensional organoids or mini-organs.